We expect that they will take place every month and will feature an organization that performs research in radiation epidemiology and dosimetry. And for today's webinar, we are very excited to have NCI featuring their research. If you're interested in presenting in a future webinar, you can contact me. You can see here my email address. And me, together with the ISoRED webinar working group, who is responsible for this series, will work with you to identify a date and time and a format that works for you. Together with opportunities to join the webinar working group, we are also looking for members to join our other working groups. You can see here the list of the working groups we have created for ISoRED. The first three are the ones that are the most active currently. The other ones have already begun their work, but they rely mostly on the in-person meeting that we hope we will host in May 2022. Of course, depending on the way the pandemic is going. The titles of the working groups, I think they're more or less self-explanatory of what their goal is and what their tasking is. But again, you can email me and we can talk more about what your interests are, and I can explain more about their goals. Today we have about 42 working group members, and we really need your support. We're looking for additional working group members. We're also looking for leads. The two working groups with asterisk have an acting lead, but we're looking for a permanent one. How do you know whether you are suitable, a good fit for an ISoRED working group? Well, if you're interested in bringing together radiation researchers to discuss studies of ionizing radiation, if you're interested in supporting capacity building and training, and if you're interested in promoting and facilitating international collaborations in radiation research, you're the perfect fit for an ISoRED working group. And again, we welcome researchers at all career levels. If you are unable to commit in a working group, but you want to start exploring, we have an open door policy, so please consider observing a working group. I would be happy to send you dates and times where the working groups are meeting so that you can take a look on what activities they're doing. You can connect with us different ways. We have a website, it's under construction. And we have other ways to connect with you, either Twitter, ISoRED, and LinkedIn. And here I have my email address. If you're interested in participating in a future webinar series and feature your organization, you can email me directly and our team will be in touch. Thank you very much. With that, I'm gonna pass the floor to Michael Hauptmann, who is going to be introducing the speakers and today's webinar. And let me stop sharing.
Yes, thank you very much, Rania, for the introduction. It's a great honor for me to be the discussant here in this seminar. It gives us a little bit of the feeling of the radiation community that we all, or many of us, experienced during meetings we had in the past, the famous ASA meetings, and hopefully we can meet again. I want to save time for the speakers, and therefore I'm only saying that I would appreciate your comments in the chat function during the presentations and after the presentations. You can raise your hands and then I will make a selection, and if you turn on your camera, we will see you when you speak and ask your question to the speaker. And this will be a selection, so any questions that are not being answered, you can send to the speaker, and I'm sure they will be very happy to answer them to you. So without any further waste of time, I want to introduce Amy. Amy Berrington is the chief of the radiation epidemiology branch. She's originally trained in the UK with a PhD in cancer epidemiology from the University of Oxford. Many of you know her. It's one of the biggest, most active departments of radiation epidemiology in the world. And she will describe the radiation branch and what the program at NCI has to offer. I can only recommend it because I once started there. Amy, please give us an overview of NCI's REB, and then we can discuss things after your talk.
Great, thank you very much, Michael and Rania. Everyone see the slides okay? Okay, great. So this is whistle-stop tour of the NCI Radiation Epidemiology Branch and Dosimetry Unit. It's a great privilege to be able to talk to you about this morning, and for then you to have a taste of the research that we do with our three excellent speakers who will be following. Slide. Okay, there we go. So the branch was established nearly 40 years ago now. Our mission is to identify and quantify cancer risks in populations exposed to a wide range of radiation exposures. We want to inform radiation protection and advance our understanding of radiation carcinogenesis. Okay, so we have three main strategies to achieve this mission, and I'm going to cover two very briefly today, which is the research and training. Communicate what we're doing today as part of our communication strategy. So first of all, our research program. So REB research covers a wide range of exposures. Here's some examples of what we're actively involved in studying now. Obviously, Chernobyl, you're going to hear about this morning. We've had long-term collaborations with RERF studying the A-bomb survivors. We have a very active dosimetry team doing cutting-edge research methods to quantify exposures. We work in diagnostic and therapeutic radiation exposures, medical workers, and also susceptibility, particularly genetic susceptibility to radiation exposure. So we have an overarching research philosophy here where we want to prioritize studies where we're going to be able to quantify risk and look at the shape of the dose response, incorporating high quality dosimetry as a top priority. That's why we have this embedded dosimetry unit, which I think is a very special feature of our group. We want to be able to identify susceptible populations. We want to perform low dose risk assessment. We want to be looking at biological specimens to provide insights into carcinogenesis. And then we also want to be able to think about the magnitude of the risks, particularly estimating it in terms of absolute risks, to think about the risks and benefits of radiation exposures. It's of course not all bad that we are still, many of us still exposed to radiation. So here's an example of some of our current ongoing field studies, things like the pediatric proton therapy that we've just initiated with Massachusetts General Hospital, long-term study of radiologic technologists, new study of nuclear medicine workers and interventional radiologists, studies of cancer survivors, including breast cancer and retinoblastoma, and a very long-standing collaboration with CCSS, looking at second cancer genomics currently. On the environmental side, and this is what you're going to hear about today, of course, have long-term collaborations with many institutes in Ukraine, Belarus, and Russia, looking at different populations that were affected by the Chernobyl accident. And of course, our long-term collaboration with RERF and the A-bomb survivors. So we want our research to have impact in the following areas. We want it to inform radiation protection, influence clinical practice, address public health concerns, and also advance our understanding of carcinogenesis. Here's an example from the last year of the publications from our group, led by a group. You can see all sorts of different research journals in different fields, all sorts of cutting edge methods, including 3D printing, exome sequencing, and advanced epidemiologic methods. So strategy 2 now, training the next generation of research scientists, and this is equally important to REB's mission. So our branch currently includes more than 20 permanent staff. As Michael said, it probably is one of the biggest groups in the world of population scientists, at least working on radiation research. including investigators, staff scientists, and a large number of trainees, as well as our all-important admin staff. We have a multidisciplinary team of epidemiologists, biostatisticians, dosimetrists, and people coming from really a very wide range of backgrounds. Diversity is a critical part of our training program. You can see here our diverse fellows from all over the world, from diverse backgrounds. This is fellows that have been in the REB training program in just the last 5 years. So the training program at DCEG more generally is something that we're very proud of and take very seriously the mentoring of our trainees. We have many formal features embedded in the training program, annual training plans, a formal mentoring committee of 3 or 4 senior scientists from across the division. We have our own Office of Education run by Jackie Levine, supported by a number of other staff that are there just to support the trainees throughout their time here at DCEG. There are many internal training opportunities across the whole NIH community, including being able to work with and be supported by a large network of NIH fellows who come from all over the world. We have funds for external training and conferences, and we really prioritize training our mentors. And so there are both awards and training provided to make sure that we're really providing excellent mentoring for all of our staff and our trainees. We're currently recruiting. So we are currently looking for a tenure track investigator who's interested in working in cancer survivorship, particularly looking at the late effects of radiotherapy. We have several postdoc opportunities through our program, mainly the one here known as the CERTA program. We're looking for trainees with expertise in biostatistics, genetics, dosimetry, and epidemiology. And there's also opportunities through what we call our post-bac program. So this is usually a 2-year program for students that have completed either an undergraduate or a master's degree to come and work for 2 years, usually before they're planning to go on to do a PhD or go to medical school. Just to finish on the broader environment of our division of cancer epidemiology and genetics, which is a very distinctive research environment. So the intramural program at NCI has internal core funding, so we don't apply for grants. And that really gives us special opportunities and creates, I think, a very special environment. It really emphasizes the need for people to work together in transdisciplinary teams. We have long-term investments in studies for going back decades. We also actually can respond very quickly as well, though, to new initiatives and new problems. And we work together sharing these collaborative resources. So this gives you an idea of the much broader community that REB is part of. And again, our trainees and our scientific staff then have the opportunities to work across the division and are encouraged to work across the division and can work on all sorts of other cancer exposures of the wide variety shown here. So that's my last slide. I'm happy to take questions now, or if Michael wants us to move on to the real talks, we can do that. If anyone's interested in opportunities, or collaborations, or coming to work with us, please contact me directly, my email address here.
Okay. Thank you very much, Amy. That was very nice, and I think really spanned the entire sort of ambition and also things that REB provides. I think the mentoring is very important. Please, could you clarify? My impression was that when you meant mentoring, that you train the mentors to be good mentors to young people at REB. Is that right?
Oh, yes, absolutely. So a strong emphasis on training the mentors as well as training the trainees.
Yeah, that's what I also had understood and what I know from my own experience. We have another question from Lydia Wilson. Would it be possible to share links for those job openings in the chat? I'm sure we can do that, Lydia, but they are also on the website, which you can find by just typing in DCEG and REB, and you will get to that website.
I'll put them in the chat in a minute.
Yeah. Are there any people who want to ask a question themselves? I don't see raised hands. This would be your chance. If not, and if Amy agrees, I would move towards the scientific talks so that we have some time here to also have some questions. I was surprised yesterday when I tried to prepare that these all seem to be really new work. There's nothing really in the literature on those particular topics. So this is really great because we learn something that's not available to others. And therefore, I want to introduce the next speaker. Thank you, Amy. The next speaker is Konstantin Chizov. I apologize if I didn't pronounce it right, Konstantin. Konstantin has a PhD in radiobiology from Russia, is relatively new in REB since 2019. And he's a dosimetrist and working on dosimetry, a very important part of radiation evidence. And he's talking about using random forest algorithms to improve the reconstruction of doses in Chernobyl after the accident in cleanup workers. Konstantin, please. Can you hear me, Konstantin?
Yes. I heard. Hello, everyone. I tried to share my screen.
Try again. It was.
Yes.
Okay. Do you see my screen?
Yes.
Click on display setting.
Yes.
On top, click select swap. Good.
Good.
You good? Yeah.
Okay. Yeah. Okay, dear colleagues, today I present my work. It's a presentation about the random forest algorithm for dose reconstruction based on classification of cleanup workers of the Chernobyl accident. So, as a result of the accident on the Chernobyl nuclear power plant, heavy contamination of the environment occurred, and thousands of workers took part in cleanup works, and only a few percent of them have sufficient dosimetry. Therefore, several methods for those reconstruction were developed, and one of these methods called RED-RU is based on the time and motion method. So it's uses layers of the those maps and routes of the personnel, and it's good. It gives good results, but it is time-consuming method because it includes long in-person interview by an experienced interviewer. The other methods were based on the expert evaluation. So if the expert knows very good situation. So based on the date of work, based on the place of work of the liquidator, he could say what is the dose for this person. But experts, they are very rare. It's hard to find a lot of experts to reconstruct doses for a large group of people. So the idea of our work was in developing an algorithm that works like an expert evaluation. And this algorithm should be based on the machine learning approach. And the neural network is being trained on the existing data, and it starts to work like an expert evaluation. In this work, we used as a train the data from RED-RU and Rockwell, which been approved in our past studies. Here is more about the objective of our work. So all information of doses of the repeaters, this is in the state registry of the Ukraine, and we took sub-register of this. It is CR registry, clinical epidemiological registry, and our aim as a dosimetry group was to develop a dosimetry system that could efficiently characterize radiation doses for this group. And as I said before, we will use the machine learning algorithm, and we will use the gold standard dose, as we call the doses which were reconstructed by the Redru method. And what we have in our database. In CR, we have about 11,000 subjects with the basic information of their work during the ChinPP cleanup works. And also we have some demographical and epidemiological data on these people. And for the test and train, that we have our past NCI studies, about 2,000 subjects with reconstructed doses, and also the demographical and epidemiological data on them. And as a neural network, we use the random forest algorithm and the code developed in our language. So our tests show that if we try to reconstruct those just using the parameters in the database, it gives not efficient result. The accuracy is not high. That's because our database has some general parameters, and it doesn't have a specific long questionnaires like in Redru, where exactly this person was during every hour of his work. Usually we have only one place of work or one place of residence during the mission. So we decided to do the reconstruction in 2 steps. And first step is a reconstruction of the contingent of the liquidator. It's a specific group of the liquidator. And then on the second step, we will reconstruct the dose inside this group, because for each group we have a dose range, and using conditional parameters we could clearly specify the dose inside this group. Okay, so these contingents, they are previously selected in previous studies. They are in basic 12 groups, and each contingent has a difference in the dose monitoring. For example, for victims and witnesses of the accident, and for liquidators, they almost have no dosimeters, because it was the beginning of the accident. And also the quality control for the military liquidators was also low. But for example, for other groups who started to work after the beginning of June, sent to mission to the fatigue control zone, they had the necessary equipment, and dose reconstruction for them and dosimetry for them was pretty better. Okay. Also, if we see on the dose distributions for each contingent, we see that the dose distribution is also different, and the form of the dose distribution could also show us how the dose was managed, because if you look in the center of this, in the center top of the slide, and in this purple cloud, there are log normal distribution, and also if you can see, not all the distributions are in this area, and some of them moved to the right, to the chapeau area of those distribution. And also some of the distributions have a short peak, for example, for the military liquidators. So it means almost all in this group have exactly one dose, but it's maybe not so correct. So how we did the random forest calculation? So first we split our questionnaires by year. So for example, if the man works for a long time, several years, we divided his missions to the each year. And in the work, worker could have a different type of the contingent during his missions. For example, on the first mission, he was an early liquidator, and then he came again and started to work in the 30-kilometer zone as a sent to mission to the 30-kilometer zone. So we divided the contingents, we divided the liquidators, and we used the step-by-step algorithm for reconstruction. On the first step, we divided the liquidator. Is he a military liquidator or he is a civilian liquidator? And then, if he is a civilian, we also did the next steps. Is he a victim of the incident or early liquidator? And if not, then we divided the other groups, other contingents into big groups. We call this supergroup 1, supergroup 2, and then we use the neural network inside this group to separate workers to each type of the contingent. And we use a Redrow-Rockwell for train of this set. And then also we use 30% of the Redrow-Rockwell database to test this algorithm. And
Konstantin, I have to interrupt you. You have about 2 more minutes.
Yes, thank you. Yes, I'm almost near the end. And great. So it's a formula what we used in the dose reconstruction. It was basic the parameter of the database, the begin date, the duration of the mission, the place of work, the place of the residence, and affiliation to the organization and the occupation. And for different steps, it has a different correctness of the algorithm. But for some of them, it was pretty high, 99%. On average, it was 75%. So the algorithm works, and then after using the algorithm, we received the graph for the CR database for which contingent each of the workers from this database. And as we see, the main group of the liquidators is the group number 8. It's sent to mission to the 30-kilometer zone. And if you look from the dose distribution for this contingent year by year, we also see that the dose range is different because the contamination levels goes down. So if we know the group of the liquidator, the contingent, and know the date of the work, the year of the work, so we can also work inside this dose range to understand the dose of the liquidator. Okay, I'm coming to the conclusions. So we use the random forest algorithm to reconstruct the doses, and it's necessary that the initial entrained data have sufficient information. That means that all the data for training must contain all variants of the data that are found in this studied data set. And we restored 9 contingents for people from CR database, and the main group was the contingent number 8 sent to mission. And the step-by-step algorithm that we have developed, it increases the accuracy of the determining the contingent from 70 to And it quite well works for the distinguish between military and civilian liquidators. And on the future step, we will continue to develop this code. We will use the other parameters from the database to develop this algorithm, and we also will do the next step. We'll reconstruct the dose inside the contingent for each of the liquidators. Okay, thank you for your attention. I hope I was in 10 minutes.
Yeah, thank you. That was very nice. This is, of course, a challenge with a complex topic like this. Let's see. I'm looking at the participants list because there was no question in the chat. Is there anybody who wants to ask a question to Konstantin? As long as there are not many, you can also speak up if I don't see you in time. No questions so far. Konstantin, the thing I'm more on the epidemiology side or statistics side. The dose estimates that your algorithm produces, with what type of uncertainty do they come?
Yes, we use statistical uncertainty, but we have received it in our previous studies in the red row. It has a algorithm for the uncertainty, what takes into the account shared and unshared errors. So we use our work based on this data.
Mm-hmm. Okay. We have a question from Kosti, who asks whether this could also be used, probably in principle, for medical research. I assume, Kosti, you mean medical radiation exposure, so treatment or diagnostics, and you would need some sort of gold standard population. What do you think, Konstantin?
Okay. Well, here I speak as a gold standard for the dose reconstruction to the GMPP accident, because it was a specific work. If you mean to use these doses and see the epidemiological consequences, yes, it works. This is work from. This is one of the part of work at NCI, because this code red rule, it was used for the leukemia studies and for the thyroid cancer studies.
Mm-hmm. Okay, thanks a lot. It's a complex topic. I'm afraid we have to move to the next speaker.
I have a question, Michael. Sorry.
I've been trying to ask the question, but I was muted, and somebody just unmuted me.
I know. And 1 minute, Elizabeth.
Okay. Yeah. Sorry. Just very quickly. Thank you very much. It's very nice, Konstantin. You mentioned something about the work regimen, so whether they're shift workers or others. I don't remember that in our questionnaires for the liquidator studies we had that much information, or that complete information, whether they were shift workers or other workers. Is there much information?
This information is in the CER database, and for Rockwell and Retro, we could extract it from the contingent of the liquidator. For example, if he works on the GNPP, this person has some specific work regime, and then based on this correlation of the work regime and contingent, can do the same correlation. If we know the work regime of CR worker, so we can understand the type of the contingent of this worker.
Yes, thank you very much. I suggest that you can discuss this further. off record because I want to save time for Elizabeth and Lindsay later. Elizabeth Cahoon is a tenure track investigator with a PhD in biostats from Johns Hopkins. She's in RIB since 2010. So for 10 years, she's working on ionizing radiation and UV radiation. And she's today talking about thyroid cancer risk in cleanup workers from the Ukraine in Chernobyl. Elizabeth.
Thank you, Michael. You're promoting me. It's a PhD in epidemiology.
Check your website.
So yes, good morning and afternoon. I'm very pleased to present today risk of thyroid cancer in Ukrainian cleanup workers following the Chernobyl accident. So as many of you know, on April 26, 1986, a fire at the Chernobyl nuclear power plant resulted in a massive release of radioactive isotopes into the environment, including iodine-131. This occurred in northern Ukraine, very close to the border of Belarus in what was then the Soviet Union. Something important to keep in mind is that since the thyroid gland uses iodine to make thyroid hormones, iodine concentrates in the thyroid. This makes the thyroid gland especially vulnerable to high exposures to this type of radiation. So following the accident, over 500,000 workers in the Ukraine, Russia, Belarus, and Baltic countries, mostly men, participated in various decontamination and cleanup operations. There were substantial and variable thyroid exposures, both external and internal, which included ingestion and inhalation. Since the accident, increased thyroid cancer rates have been reported among cleanup workers when compared to the general population in Ukraine. And those findings use a standardized incidence ratio approach, and there were some concerns for detection bias due to screening programs and increased awareness of radiation-induced cancer. So I think it's safe to say that high risk of thyroid cancer from radiation exposure during childhood is well established. External exposure has been studied among the atomic bomb survivors and medically exposed populations. Internal exposure has been studied among populations exposed to fallout, such as the Ukraine and Belam cohorts. However, there's considerable uncertainty for radiation exposure in adulthood, especially among men. So this is an important concern for both environmental and occupational exposures, especially among the public and for nuclear and emergency workers in the case of another nuclear accident. So a case control study of 107 thyroid cancer cases, which included 66 men and matched controls among Chernobyl cleanup workers residing in Belarus, Russia, and the Baltic countries, showed a strong dose response among men as well as women. And among the male Japanese atomic bomb survivors who were exposed at age 20 years or later, a non-significant negative EOR was reported for thyroid cancer. Most of the other studies of medically and occupationally exposed adult populations report positive risk estimates, but many of those may have limited power because of either low doses or small numbers of cases, particularly among men who have generally lower background rates of thyroid cancer. So given these limitations in many of the studies, an independent assessment in cleanup workers was needed. So the objective of this study is to examine the association between thyroid radiation dose received during adulthood and thyroid cancer risk. We conducted a nested case control study among male cleanup workers included in the Chernobyl State Registry. About 3 controls were matched to each case on year of birth, old list of residence, and survival at time of thyroid cancer diagnosis for the corresponding case. Thyroid cancer cases were ascertained through the Ukrainian Cancer Registry. And the study includes the largest number of cases of any study of Chernobyl cleanup workers with individual thyroid radiation doses. So individual thyroid doses due to external and internal exposure during the cleanup mission and during residence in contaminated settlements were calculated for all the study subjects. External radiation came primarily from occupational exposure during the cleanup mission and was based on exposure rates through the 70 kilometer zone and the whereabouts of the liquidator. Personal interview data was collected on duration and location of cleanup activities, and also traveling and resting during the cleanup mission. Internal radiation from occupational sources included inhaled iodine-131 and a number of short-lived radionuclides. Internal radiation from residential sources included iodine-131 intake with locally produced milk and other food. Most subjects received doses under 100 milligray from all exposure pathways combined. The median contribution of internal radiation in this low dose group was about 60%. And the total dose mean was 199 milligray and ranged from 15 milligray to 9 gray. Actually, I think I'm 20 subjects received thyroid doses of 1 gray or higher. As you can see on this histogram with the darker bars, doses in this group were mainly due to external irradiation. And you can find more information about this in the publication last year in Health Physics. So here are the main overall results. Although category-specific risk estimates were not significant, the odds ratio was the highest among the highest dose categories. The excess odds ratio per gray was 0.44 for total dose after adjusting for proxy, smoking, and alcohol. And the dose-response relationship was consistent with linearity when we fit a dose squared term. We also examined the radiation dose response according to some selected characteristics. The EOR per gray for internal dose was 0.39 and 0.47 for external dose after mutual adjustment, so pretty similar to the results for total dose. We did not detect modification of radiation and thyroid cancer risk by use of stable iodine while on mission, by attained age, or a number of other characteristics not listed here, including oblast and age at first mission. Time since first mission modified the dose response associations with borderline significance, so risks were higher for the 0 to 15 years since first mission when compared to 16 years or later. We also examined the radiation dose response by tumor type. The EOR was highest for the follicular tumor for follicular thyroid cancer, and it was borderline significant. The EOR was lowest for papillary thyroid cancer, though you can see that the confidence intervals were overlapping. And these results were not sensitive when allowing for a 5-year latency. However, among the papillary thyroid cancers, less than half were diagnosed within the 15 years of first mission. And in comparison, 75% of the follicular thyroid cancers were diagnosed during that first 15 years. So since the follicular thyroid cancers tended to be diagnosed earlier, it's challenging to really separate the effects of time since exposure and tumor type. So in summary, total thyroid dose was associated with increased risk of thyroid cancer with borderline significance, with some evidence that the magnitude of risk varied with time since exposure. There was also borderline significant but marked elevation in risk for follicular thyroid cancer based on a small number of cases. I think these findings are generally consistent with the growing evidence of an increased thyroid cancer risk observed in most studies examining radiation exposure during adulthood. Though the magnitude of the effect in this study is lower than the previous case control study of Chernobyl cleanup workers.
Yes.
I'd like
to... Yeah, go ahead. This is important.
So, I'd like to gratefully acknowledge the friends and colleagues involved in this work. This research was supported by the Intramural Research Program at NCI, and it was led by Senior Epidemiologist Dr. Natalia Gudzenko at the National Research Center for Radiation Medicine in Kiev, Ukraine. And I'm happy to answer questions if there's.
Okay. I don't know if others could hear the acknowledgements, but we could see them. I couldn't hear you during that minute. Yes, very good. Thanks for a very nice presentation, also for being on time. It's really a pity that we have to rush through this quite a bit. And I would really like to give somebody the opportunity to ask a question in person, because reading from the chat is second best. Let's see if I can find anybody. If you have one, please speak up. And I don't see that. Okay, then From the chat, one easy question is whether the doses are effective doses or equivalent doses on slide 9. So you calculate thyroid doses, which reflect external and internal exposure. Can you speak to that?
I think that's a question for the dosimetry group. I don't know if anyone is on here. I think that would really best be answered.
Okay, or if nobody's here, you can try and find out and send the answer to Marie Cruz Caruso. And there's a question by Lydia, whether the risks by intake of stable iodine seem to be reversed. Lydia, would you feel okay to ask your question directly? Okay, that doesn't seem to be the case, or she cannot unmute herself.
I think that's the problem. So Abigail, if you can unmute Lydia Zablowska, please. For some reason, I cannot do it right.
Yes,
Thank you very much. I apologize for wasn't able to unmute. This is really great presentation. Thank you so much, and I'm happy to see the results. The quick question is, on one of the tables, there was a heterogeneity of estimated risk by intake of stable iodine, and they seem to be reversed and showing higher risks for those who took it. It was above 1 against those who didn't take it, and they had negative risks, I believe. Do you have any ideas why that could be or what's happening?
So in the model for both of the internal and external doses, those were both included at the same time. Mm-hmm. So, and I felt like the risks were very comparable. You know,
right, because they were not statistically significantly different. Okay.
Yeah, they were not significant, and the magnitude of the risk was similar between the two and to the overall total dose EOR.
Thank you.
Yeah, this is always hard to say with a relatively small data set. I mean, yours is the biggest so far, but is still small when you slice down the dose response into subgroups. Something many of us have experienced. Anyways, thanks a lot, and again for the nice presentation. And I want to reserve at least 10 minutes for the last talk, which is by Lindsay Morton. Lindsay is the deputy chief of REB, so it's an honor that these two younger people are surrounded by the chief and deputy chief. Lindsay has a long track record of working with in REB. She has organized great consortia. I had the pleasure to be part of one on second gastrointestinal tumors, and I think she's really has a talent to do the science, but also to bring people together. And I think the topic that she will talk about is also a very big effort of bringing people on diverse disciplines together. Lindsay will talk about molecular topic on thyroid cancer in people exposed in Chernobyl. Lindsay, the floor is yours. Lindsay, can you hear me? I don't see Lindsay in the list of...
You're muted, Lindsay.
Okay, is that better?
Yes, much better. Thank you.
Okay, great. So, sorry about that. I thought I had unmuted and then I got remuted. So thank you, Michael. Exactly as you had said, it's really a privilege to have the opportunity to present these results today, which represent the work of a very large interdisciplinary team of investigators. And you had also mentioned it's hard in these webinars. We have a short period of time, but our goal was to, of course, have Amy present an overview of the research in our group, and then Konstantin, Lisa, and I to have the chance to just give you a flavor of the range of research and present some new data. So we're very happy to take some additional questions, knowing that these presentations were all short. So I'm going to go through and kind of skip through the background a little bit in the interest of time. Of course, we all know that the most important residential exposure following the Chernobyl nuclear power plant accident was from internal intake of I-131, and that there is an established increased risk of thyroid cancer with increasing radiation dose, particularly for individuals shown in the dark line here who were exposed at younger ages. So when we think about the molecular background of papillary thyroid cancer, it's known that there are specific mutations in papillary thyroid tumors that can influence therapy. There's a most comprehensive landscape paper looking at the molecular profile of sporadic thyroid tumors published by the Cancer Genome Atlas, which showed that thyroid cancer has a very low mutational burden, but that increases with increasing age, and that the frequency of fusion drivers decreases with increasing age. So fusion drivers are more common among younger individuals. A nice RNA-seq-based analysis of post-Chernobyl papillary thyroid cancers also showed the fusion drivers are increased with increasing radiation dose. So it was set against this background that Stephen Chanik, in partnership with the Chernobyl Tissue Bank led by Jerry Thomas, and collaborators from the Ukraine, led by Mikhailo Tronko and Tanya Bogdanova, set out to comprehensively characterize the molecular profile of 440 fresh frozen, pathologically confirmed thyroid tumors occurring after the Chernobyl accident. And the aims of the project were to define the spectrum of driver mutations in these tumors by radiation dose, and then characterize the profile more broadly, looking at the mutational, transcriptomic, and epigenomic profiles, and again, relate all of these molecular characteristics to the radiation dose, which enabled us to ask questions like, is there a biomarker for radiation-induced cancer? And when in the carcinogenic process does radiation have an impact? So among the 440 tumors included in our analysis, the mean age at papillary thyroid cancer was about 30 years. We had 81 unexposed individuals, and then the mean dose among the exposed was 250 milligray. And you see here in both the dark and the light gray bars that the majority of the tumors developed among individuals who had received less than 200 milligray. So to do a genomic landscape analysis, we used a range of different laboratory methods listed here on the left. And we created 68 primary analysis variables, looking at single nucleotide variants, small indels and microsatellite indels, structural variants, the driver of the tumor, somatic copy number alterations, and then a number of different patterns looking at the transcriptome, as well as germline variation. This is a complex analysis with a lot of different parts. I'm obviously not going to tell you all about it in this limited time, but wanted to give you a sense of the scope of the analysis. And what I'm going to focus on today is just a few key results where we identified those molecular characteristics that were strongly associated with radiation dose using multivariable regression models that were adjusted for age at thyroid cancer and sex. So this is a plot of the typical landscape type analysis, and in the interest of time, I'm going to actually skip that and go to the main results. So we identified an increased number of deletions with increasing radiation dose, as well as an increase in the deletion to SNV ratio. So this ratio gives a sense of controlling for the background noise in the tumor. And this association was even stronger than just looking at deletions alone. But in contrast, we saw no association for dose with single nucleotide variants or insertions, whereas all of these variants were associated with age at thyroid cancer. So this gets back to the importance of the multivariable model. We looked at structural variants, and this Nature paper by the Pan-Cancer Analysis Working Group, or PCOG, is a valuable reference to learn more about structural variants. But briefly, this gives you a sense that we have both simple and complex variants. And in particular, we focused on simple and balanced structural variants, so those that don't result in a copy number change. What we found was that increasing radiation dose was associated with an increased count of any type of structural variant, but particularly those that were simple and balanced structural variants, whereas there was no association for dose with complex structural variants and only a marginal association with simple, unbalanced structural variants. Moving on to the driver, because we had such a comprehensive set of laboratory studies, we were able to identify a driver for nearly all the tumors, and almost all of them were drivers in the MAP kinase pathway as listed here, with 253 mutation drivers and 176 fusion drivers. We found that increased dose was associated with the increased likelihood of having a fusion versus mutation driver, so this was consistent with the previously reported RNA-seq analysis. But there was no association for dose with the specific driver gene that was either fused or mutated. In terms of germline variation, I'm going to go through this pretty quickly, but I'm happy to take questions. Based on a polygenic risk score looking at common genetic variants, we found that individuals with higher radiation doses actually had lower genetic risk. So as we summarize the primary analysis variables, I put in red here the key molecular characteristics that were associated with radiation dose. And for each of these characteristics, we did a whole range of secondary analyses. And I want to just focus on two here. One is to look at the clonality, and the other is to look by age at exposure. What we found when we go back to the deletion finding, and the results were also consistent for structural variants and the fusion, so the driver type, was that this was the association I showed you previously for the total population. But the associations were much stronger when we restricted to clonal events as opposed to subclonal events, which provides evidence that the radiation has an impact early in carcinogenesis. We also saw a stronger radiation effect at a younger age at exposure. So it was really interesting and exciting to see the molecular data parallel the epidemiologic data. So in conclusion, I'm going to jump down to the bottom here. Our results provide evidence that environmental radiation exposure causes DNA double-strand breaks early in the carcinogenic process that subsequently enable papillary thyroid cancer growth. And this integrated analysis was a really exciting opportunity to take a typical landscape molecular profile analysis and combine it with an etiologic factor like radiation dose. In order for this work to be done, of course, it took a tremendous interdisciplinary team, and I'm really grateful to all of our colleagues for all of their contributions, and I'm happy to take questions.
Thank you, Lindsay. I was initially afraid that your 20 slides will mean that we will, take much longer, but this was perfect. Thanks a lot for a very nice talk, and let's see if we can have one person that asks a question in person. That would be really my goal, because that's also part of the exercise here. I see Anthony waving. Let me find you in the list and unmute you, Anthony Brooks. Request unmute. Rania, can you unmute? Yes. Please ask your question, Anthony Brooks.
I really enjoyed your presentation. But it looks like the work we've done 50 or 60 years ago showed exactly the same thing that you showed as far as chromosome deletions, etc. They're all good markers of radiation dose, but poor markers of radiation risk. I wonder if you'd comment on, do you have anything that's really a marker of risk, or all these just markers of radiation dose?
Yeah, so that's a great question. I apologize that my video is no longer working. So I think there are two elements to your question. One was we indeed were very excited to see the consistency of our results with a lot of the previous literature. What was exciting and novel about it was the ability to go to really comprehensively profile the molecular characteristics of the tumor. And so that's never been done before with fresh frozen tissues available from the Chernobyl tissue bank. It enabled us to look at a lot of different characteristics and address a whole range of other questions, including things like cluster damage and things like that, which I didn't present in the interest of time here. I agree that we cannot measure risk. So these are all metrics of dose. I think what's novel about the findings is that it provides insight into how radiation causes cancer, consistent with a lot of data that have been shown previously, but not actually in human tumors. So not a risk metric. And also, to elaborate, not a unique biomarker. So there are other carcinogens that can cause DNA double-strand breaks. So there was not a unique radiation signature in our analysis.
Thank you.
Thanks, Lindsay, for this answer. I think there would be a lot more to say about these data, and while it gave us a very good summary, there's, I guess, a lot more in it. I want to close the discussion here because it's 4 o'clock where I am, and I know Amy said good morning in the beginning, but we have people from many continents, many time zones. From my point of view, it was a pleasure. I want to say that the videos will be available on the website of the ISoRED Society. As Ourania said, we are looking for volunteers to feature their institute and their research in future webinars. And I want to thank Ourania and, of course, everybody else in the ISoRED webinar group for organizing the webinar. And I hope you will all be back for the next webinar. Ourania, do you want to close the session?
It was well said, Michael. Nothing else to add. Thank you to everyone for participating and for presenting. Have a good morning, evening, afternoon.